Welcome to this video 3 of G01, our short course on read library quality control. And now we're going to look at read library types. So there are several different read library types, and we're going to mainly focus on two major types in this particular article and video. So what we need to bear in mind is how read libraries are generated, and we're not going to talk about the technological details here. We're going to bear in mind that we have some DNA of interest. We fragment it to create the reads. We then sequence them using a particular technology where they're amplified in a particular manner that's specific to that kind of technology. So for Lumina, that could be sequencing by synthesis, but we should bear in mind that the other chemistries are quite different as well. And then we can do downstream analysis afterwards of whatever form. We have to check that our reads are actually good and that they pass quality control first. So the outputs of different technologies varies a lot. There's many technologies. The Illumina, we mainly know that they're associated with GA2, HiSeq, MiSeq, NextSeq, NovaSeq, and so on. And then we also, for long reads, we mainly have PacBio and Nanopore. So we can kind of see the read lengths on a log scale versus the gigabases run for a slightly old diagram for 2016. And what you can see here is that we have nanopore mini-ion devices here where their output is gradually increasing. Even then, we have PacBio here that are increasing their output too. Some of these technologies no longer exist. But then you have HiSeq up here where the read lengths are shorter, but the output is relatively higher. Whereas long reads are over here, fewer reads for longer ones. And we can also see a more updated picture here of the different technologies. 454 is gone. Lumen is still here. Life sciences is effectively gone. Iron Torrent is not useful in my experience. And there's other ones that are really not really worth noting anymore. We have PacBio and Nanopore there, all of which generate useful long reads using different machine types. So results will differ even between the different machine types. So you can see here, there's a range of options, and we know at the per byte we have our HiSeq, NextSeq 500, 550, NextSeq 2000, Nanopore mini-ion, and then also we have also external sequencing at times where the data types might be quite different. So ultimately it breaks down between short illuminated reads and longer reads, typically Nanopore. So the steps used generally to generate such libraries involve DNA fragmentation and ligation on of the adapters and barcodes specific to each sample. And then we have the read library preparation, and that could be for single end reads, SE, or paired end reads, where each read has a pair from the same DNA fragment. We then have read library QC, and then the downstream analysis. So what we do here in terms of QC and downstream analysis depends on the read library type. So single end and paired end are treated differently for certain steps. If we look at the structure of DNA reads, we know that, of course, they have the fragment here, the DNA fragment from which they come here in the middle, and that can often be quite long, maybe 800 or 1,000 bases, depending. We then have the adapters that are shorter, and then the barcodes that are shorter again, usually. So you note those barcodes at each end and adapters at each end as well. If we're dealing with single end data, there'll just be one sequence read corresponding to the 5' end of the DNA of interest, and we'll have a primer on here that will allow the DNA polymerase to extend across this read to make a complementary sequence to this insert. If we have paired end reads, we'll have two reads together, where the second read that's known comes from the same DNA fragment. So these two read pairs face each other with some unknown insert size here, I. In the middle, that might be 300 to 500 base pairs, depending on the library preparation approach. These reads here might be anything from 100 to 150 to 300 bases for short read data. For long read data, we only have single end reads only. So note those differences. There are more diverse read library types beyond just these. So we have single end and paired end reads we talked. We also have mate pair and also linked reads as well. So if we look up here, it's possible to make pair library, which essentially have read pairs shown here and here, where the distance between them is very, very long. So that's very, very good for scaffolding, but not necessarily good for getting a large number of reads. We also have single end and paired end, as you mentioned before, and then linked reads or stroke reads or the different names are also sometimes used. For us in this practical, we're going to focus on two types essentially of Illumina, which are short, numerous, and accurate reads. And in contrast, we have Nanopore, which are longer, but fewer and less accurate reads. So we can summarize Illumina using this diagram that they're short, they're very high quality, typically paired end if useful. For many viruses, it's not actually worthwhile even doing paired end. It depends on the approach. They are PCR, so you may get a lot of duplicate reads, and you have to take some care for looking at that, especially if it's a virus where amplicon sequencing has been used and you get very many of them. So this PCR effect can have a substantial difference to the output, and we have to bear in mind that we're trying to resolve each base using these reads. For nanopore reads or PacBio indeed, they're very, very long. Their quality is decent. It depends on the chemistry, again, that's used to apply. It can be good. It just depends. Older samples, like some of the ones we look at, you can see the quality is definitely lower. They're single ND reads, there's no partner. There's typically less PCR involved, but there might be amplicon sequencing for viruses. And typically, there's far fewer of them compared to Illumina data. So they're very good for scaffolding the genome and getting the general structure and layout and proximity and order of genes, but not necessarily good for base resolution. So these ideas behind read quality scores are not new. They've been around since 1998, where those two papers by Ewing et al. And Ewing and Green, where they looked at the FedFap concept pipeline. And that was used for looking at genotype data, where people like me, we used to eyeball genotype data, and we were looking mainly at the base quality scores and the extent to which the chromatogram, the peaks for ACGT were sufficiently strong to distinguish one exact base only. And they developed this base quality score scale, where if Q is a value equal to minus 10 by the log base 10 of P, where P is the error probability for that call. So if you transform P and Q is 30, P corresponds to 0.001 and so on. So if Q is 20, is 10 to the minus 2. If Q is 30, is 10 to the minus 3. If Q is 40, is 10 to the minus 4, and so on as the error rate. So in that way we can transform known base quality scores into the error rate. For nanopore data, the score is calculated slightly differently. We have the fraction of the bases that matching the reference of all bases is given by the number of matches divided by all our other match types. So the number of matches, mismatches, and the number of indels too. So if we have two alignments here where we have this low quality read here on top and the reference there, on top and the low quality read on the bottom, see that there's 5 deletions and 1 insertion there. So we can compute the score. And we've got these 5 deletions, 1 insertion, 1 mismatch. So that means we can compute a score of 86 in this particular instance. So that's how we can compute quality scores for nanopore data. If we look more broadly at where QC lies in this bigger area, we can see that it's very fundamental. So this is looking at PacBio data here on the left. So you can see that once we pass QC, once we're doing QC, we look at read polishing with cover or arrow after polyA trail trimming with IsoSeq3. Another option over here for nanopore data is looking things at 4-CHOP, nano QC, nanostat, and other options here. And that's before we go into other advanced methods like read error correction, which we'll mention later, and then de novo assembly and so on. So QC is one of the key steps, as is read error correction, alignment to a reference, de novo assembly, assembly finishing, read mapping, metagenomics, and so on and so on and so on. So this diagram kind of gives you a sense of an overview of things that are used in this area. So we're mainly looking at long reads here, or the technologies that produce them in nanopore and PacBio. We're going to look at nanopore reads here, and we know there's many tools to look at them. So we're just going to look at a few for the long reads and the short reads to give a sense of where they actually lie. And let's also note that there are certainly sequencing costs, but also costs associated with computational analysis can often be higher than those for the original read library generation. With this workflow that we have our experiment, sequencing, our raw reads, intermediate files, our interpretation of results, and so on. But now, sure, the sequencing is expensive and there's costs in kind of sample collection and all that sort of thing, and data management and downstream analysis. But in the future, what we'll find is that the setup is expensive and then the downstream analysis is expensive, but the other parts are not quite as significant. So what we want to do is then to be able at the end is to take our reads and map them to a reference like the blue one shown here. We have our black reads are mapped to this reference as shown here. So that concludes our video number 3 on read library types. So please see the next video on FASTQ file formats.